So hello, everyone. My name's Helen Rosenfeld. I'm the Executive Director of the Massachusetts Science and Engineering Fair, and I'm so pleased to convene this wonderful group today. MSEF was founded in 1949 by the American Academy of Arts and Sciences alongside MIT professors and collaborators of high school teachers who created the first statewide fair, one of the first in the nation. And the fair is just the culmination of a year-long independent research project. Students are taught, they're coached, and encouraged to persevere in scientific research and engineering design, working on their own and guided by a teacher and sometimes mentors. And they're tackling real-world issues such as climate change, finding a cure for Alzheimer's, using tech to help with those physical challenges. And like professional scientists, our students are creating hypotheses, they're analyzing data, they're learning from their mistakes, they're making adjustments, and they're trying again and again and again. We believe that nurturing curiosity and encouraging independent research can lead to great things, both for the kids who take on these projects, as well as for the communities that will ultimately benefit from that innovation. So we do our work in a variety of ways. We are known, of course, for the fairs, and they recognize students whose research has earned them recognition from our esteemed volunteer judges who prod and assess their research, but indirectly also serve as role models for their future pursuits. And it doesn't hurt that there's significant scholarships and prizes that are awarded. So we also support educators across the Commonwealth with a variety of professional development offerings and as well educational tools that aid them with supporting inquiry-based learning. And we work with school districts. Today you'll hear from our Worcester partnership to run middle school STEM clubs that connect local volunteers from industry and academia to the classroom. And this exposure is so key to this week's theme, Mass STEM Week's theme of See Yourself in STEM. And we're launching a coaching program for students of color to ensure that we have a diverse representation of the Commonwealth at the regional and state competition levels. We're extremely thankful for the deep investment of area corporations, foundations, and academic institutions. And so I'm so pleased to have Arthur Heinricher with us today. He's the Dean of Undergraduate Studies and a Professor of Mathematical Sciences from Worcester Polytechnic Institute. WPI has supported MSEF and particularly the regional middle school fair for many years. And for this, we're so grateful. So I now ask Dean Heinricher to share a few remarks.
Okay, thanks, Helen. I was asked to answer the question, why does WPI support the science fair and why will WPI continue to support the science fair? To me, this seems like a pretty simple question. And I can answer in a couple of ways. One, to the students, fundamentally, you are the future and you are our future. Some of you will be students at WPI, some of you will be students at other colleges around the country, but you are the future students for all the colleges in this country. I also want to argue that math and science and engineering are really all about clear thinking, and clear thinking matters. Science matters today more than ever. You are living in a time that proves the importance of good science. You see it every day. But I want to touch on two other points that matter, at least to me. Science fairs teach you about the value of failure. I think Helen mentioned that already. I'm not going to claim that failure is good. I might argue that not all failures are the same, but I will claim that failures are part of science, and I'm confident that many of you will have wonderful failures along your journey. Learn from the failures. When you fall, and you will fall forward. Long ago, 20 years before the first science fair, and about 100 years ago, a famous mathematician, Alfred North Whitehead, wrote an essay on the aims of education. And I go back to that essay basically quite often. He talked about the purpose of the university. And I think too often we think and have thought that the purpose of a university, the purpose of a school is to convey knowledge. Teachers transmit, students receive. Whitehead argued that the real purpose of the university, the real purpose of education, and the real purpose of every project that you will be presenting at the Mass Science and Engineering Fair is to bring together imagination and experience. Students bring the imagination, that's your role. Teachers and mentors, you bring the experience, that's your role. The projects don't work if you can't bring those together. Education doesn't work if you can't bring those together. That combination is magic. That's where the future is built. And this magic is why WPI will always support the science and engineering fairs. I just wanted to say thank you. I'm happy to join this meeting today. And Helen, it's back to you.
Thank you so much, Dean Heinricher. Now it's my pleasure to invite Lynn Burns and Jen Felicetti-Magsino to have a conversation with us. Each will introduce themselves, but Lynn works with a company in Worcester called AbbVie, and Jen Felicetti is a teacher at Worcester East Middle School, and they'll speak to the interesting collaboration that they have between the school and the company and how important volunteerism and mentorship is. But I will hand it to both of you to share that in more detail.
I can go first since I'm already unmuted. So my name is Lynn Burns and I work at AbbVie. I'm the lab manager and a senior scientist in a preclinical safety group. And I've been there 6 years. I started mentoring with Jen's class 3 years ago. Yeah, it's been 3 years.
Yeah.
And yeah, it's been amazing. I guess we're going to talk more about that. But Jen, you want to go ahead?
Sure. So I'm Jennifer Felicetti, and yeah, I've been teaching for over 20 years. I'll leave it at that. And I started at Worcester East Middle 3 years ago and was blessed to take on the Science Fair Club. And when I did, I was also blessed with the opportunity to have volunteers enter my classroom. And that's when I met Lynn. So Worcester East Middle is a pretty unique environment in that we only serve 2 grades. So we have 7th and 8th grade, and we have about 750 students, and about 80 of those students are poverty level students. They qualify for free lunch. They qualify for assistance in some way. They're often first generation Americans. They will likely be first generation college students if all things go well. And they love the scientists. When the scientists do come or when they're scheduled to come, I will hear school starts at 7 and we wrap up around 2 o'clock. And I will hear pretty much from 7 to 2, students will pop into my room all day. Are the real scientists coming today? Are the real scientists coming? So we've been really blessed to have Lynn there right from the beginning. And yeah, let's see, what else? Lynn, you want to tell us from your experience? You walked into the room and you met this really hyper stressed out teacher.
I don't think I knew what I was doing either at first. So I'm pretty much just as excited as the kids are to all day leading up to going and mentoring. I grew up in New Bedford and I went through the public school system there. So I went to a school a lot like theirs. So it's really awesome to be able to kind of come in and like give back to kids that grew up a lot like I did. They're amazing. You know, to me, science is all about, it's so funny that you use the word magic, because I used it earlier this week in sort of describing what that feels like when you come in. And for us, I think it's a lot of asking questions, right? Because science to me is that curiosity. So what I see my job as, you know, being a mentor is to sort of come in and spark that curiosity. And that's the magic of it to me, is sometimes I remember being a shy kid and I didn't like to, I loved science, but I didn't talk too much about it. So to know you're surrounded by a group of people that love science, and then to sort of like pull it out of the students and get them involved, that's kind of the magic of the program, I think, for me.
Yeah, absolutely. So many of our students are, let's see, I believe we have 60% Latino, 20% Caucasian, roughly 20% Caucasian, 20% African American, and 8% Asian American. So we've got a pretty broad range of folks who don't look like me. And it's important to me to try and get volunteers to try and have the students see themselves, like you had said, you know, just these are the future. these students are our future. And if they don't see a broad future for themselves, it becomes quite narrow. So the idea of seeing themselves as a scientist is really, really great. If I may, Lynn has tattoos, and there we go. And the students are really funny. They'll just sort of, I can see them eyeballing each other, doing the chin, the chin, look over there, and they get all excited. They won't say anything. They're polite enough not to say anything, but it's very obvious that they're noticing. And after the scientists leave, they'll say, "She has so many tattoos," and I say, "Yes, she does, and she's a scientist." Yes, they can be synonymous. So it's pretty neat, and it's pretty not, I tend to ask for folks who look like my students, because I do want to see them to see themselves in these roles. They are, as I say, we have a high level of poverty, a high level of students who qualify for free and public lunches. And they are often, their families are a bit overwhelmed by maybe meeting the needs of their kids. And so having someone, an adult, who can spend the time with these students and pull some knowledge out of them and show some interest in their intellect is really neat for them. They don't have that opportunity. And to have a small group setting, as a teacher, I'm in a classroom of 28 students often, and I'm sort of managing, not sort of, I am managing all those different personalities and all the work. So I might not have the opportunity to connect with the students the way that our volunteers can.
I'm going to hop in and ask a question.
Okay.
So can you speak a little bit about the difference between what happens in a science classroom and what happens in a STEM club, and the challenges, of course, of covering content that's required for science, the requirements of a school day, with what happens in this environment where it's by choice, often in the after-school setting?
Sure. Let's see. There was sort of a multi-part question there. So I'll start with the end and work my way back. So the choice. Oftentimes, what we find as a challenge is that students do not have the opportunity to expand their own interests, because we have such a tight curriculum. And the choice part of that can be really interesting, because we have low-income kids, because we have kids who are juggling all sorts of responsibilities at home. They may not have the ability or the opportunity to be as involved as they might want to be with their academics or any sort of extracurricular activities. So in the classroom, we are, go, go, go from the time the kids get there until they leave. I need to keep them engaged. If I don't, then there is anarchy, because it's middle school. And there are days that, Lynn shows up and other volunteers show up, and I'm so grateful because I'm genuinely exhausted. But the curriculum doesn't allow for a lot of independent work. It doesn't allow for a lot of exploration. And the kids who are able to manage to get to our after school program, they've got to take care of transportation home. They've got to take care of trying to navigate their homework, as opposed to perhaps taking care of family members and things of that nature, younger siblings, older parents, whatever, grandparent care. So they're given this opportunity, if they can come, to do something that they're interested in, and they don't get that opportunity very much. So it's a very different environment in that regard. It's also a lot smaller. So the scientists can really get to know the kids can get to know the scientists. The kids are able to be heard. So whatever questions they have can be answered right away, instead of we'll get to that later. And then more times than, I'm afraid to admit, more times than not, we may not get back to that question, or it might be the next day I'll say, "Oh, didn't you ask that?" And by then the thought is gone. So having someone else to answer those questions is very welcomed by our students.
Thanks. And Lynn, can you talk a little bit about the breaking of the ice, like the process of getting to know students and being from the outside, somebody they don't see in their day-to-day, and how that can work or how it's challenging sometimes?
I don't have much trouble with that because I'm kind of loud and bubbly. I wasn't like that when I was in middle school. I was very shy in elementary and middle school, a little less in high school, totally different in college. So I can kind of relate to some of the more quiet kids too. I think when I go in, I usually try to make them laugh a little bit. And then it's all for me about when we start like picking the projects, you get a feel for what their interests are. And if you can remember kind of what they're interested in, I think when you come in the next time and you ask them about like where you left off, they seem pretty amazed that you actually like remember what they were talking to you about last time. And I think that part of it, once they see you're vested and you're excited, and like we talk, so a lot of times we'll talk about where I went to school and stuff like that, so like they know I went to a school just like they did. And like I never got to do a science fair, so we can talk about like I would have been very excited to do a science fair, but it was just never put in front of me, so I never did it. So that part of it, I think that's the most fun, and it's really cool. One of our there's 3 of us, so usually we try to schedule 2 mentors to come every week. And one of our mentors actually went to Worcester East Middle School. So I know for her, that's always like really cool, is she went to the same exact school, I don't know, probably, 20 years ago or whatever. So you just have to find those little ways to connect, figure out what they're excited about. And I always felt nerdy that I loved science. So to me, it's always important. Like I never wanted anyone to know that about me because it was like not cool, So to try to engage with them and be like, it's okay. Like we can talk about this here. Like we all think this is cool and not nerdy and fun. And you can do a really cool, impactful job someday because you're interested in this. So.
Can you speak to sort of the corporate interest in doing this, and is it difficult to get people to volunteer, and sort of the interest of AbbVie to do this? Is there sort of a broader goal behind having employees involved?
I'm not sure from their end what their opinion is on that. I know someone found out about this opportunity and sort of brought it to us, and I was interested. But from my point of view, I think that I always kind of lived in Worcester, right around Worcester. So I already have like a connection to the community in Worcester. I do community service in Worcester. That was already there for me. But when you're in a corporate setting, people come from, let's say, up to an hour away, right? So they come into Worcester, but they don't necessarily feel connected to Worcester. And I think once we can, through mentorship, you really establish this personal connection to these kids, right? Like I love these kids. When we couldn't finish our project last year, I was devastated. You know what I mean? Yeah, because I'm not going to see the 8th graders again, and they didn't get to finish, and their projects were amazing. So I think that personal connection to your community, we can do a lot of good as corporations, right? Because we can bring our experience and knowledge, and like we have, corporations have internships and things like that. So, you can tell the kids these things. And then I think once like you get connected to that community and the people in that community, it just drives all of us to do a lot more. And I know there's tons of corporations in Worcester that are vested, that do a lot, but it's kind of like once you get in there and you see how amazing these kids are, like you're not going to stop mentoring, what I mean? So I think that's the real benefit is like building that empathy, and really getting vested in where your company is. And when you got 800 people on site, that's a huge impact you can make in a community that needs it.
Well, and interestingly, if I can piggyback on that, the AbbVie volunteers they're only there for maybe an hour and a half, maybe 2 hours. So they're often leaving in the middle of their workday and going right back to work. The kids, of course, will ask and ask and ask questions, and they'll say, "No, really, I need to get back. I have a meeting, in an hour." And so it's really not an enormous amount of time. It's not an enormous contribution or taking away from, their daily lives in any way. And the joy that it brings these kids is immeasurable. Yeah.
If either of you were to share what more you want out of this kind of exchange, or like, I guess, very different perspectives, but as a teacher, Jen, what else should we as partners in this endeavor be striving to provide to make this even more accessible to students?
Oh, well, I'd love to see even more volunteers. And certainly, those of color, obviously, I'm looking for that because my students are, but we'll take anyone. I'm not going to turn away anyone, certainly. Um, Folks who want to be involved, you'll find teachers are very, very open to the idea. After school is easiest. During school can be really a challenge, but after school time, like we've got set up, is just a wonderful experience. We have students who have come back and told me that they now want to be scientists, where, before they didn't, I don't know that they really knew what that term meant. But, talking to Lynn, talking to the other two scientists, it's really piqued an interest in them to learn more, to research more. So that's really, really wonderful to see. And our students are, it's great for them to have the opportunity to discuss something that they have intellectual curiosity about. And they don't have that opportunity at home. They may have parents who are working 3 jobs to make ends meet. And, their parents might be just as interested, but they're tired. So having an adult who can be a go-to, who can show interest in what's going on here, again and again. And like Lynn said, bringing that topic back, showing that they're being heard, really boosts their confidence. And I see the children sort of stand taller in class. They're more comfortable raising their hands. They don't feel as nerdy, like Lynn said, they find their people, and their people are each other. And then, of course, they're adults too. So I think it's very much a win-win situation.
Yeah, I think for me, I'd love to see more corporations and more mentors. I think I've interviewed a number of people, and I know when I was first out of college, I was not polished. I didn't sound, I don't know, I sound like I do, you know what I mean? I don't sound fancy. I don't sound like I have a lot of experience. I think depending where you grow up, like you just grow up how you grow up, and it doesn't mean you're not a smart person, you know what I mean? And I think when you mentor and get out into the community and understand that intelligence doesn't look a certain way, it doesn't sound a certain way, it doesn't matter if you grew up rich or poor or in between, intelligence is intelligence, and we have to be open enough for when we get kids in, especially right out of school, right? Mm-hmm. To realize that, picking a kid because they're the most polished or because they have 59 million internships. I worked all the way through college. You know what I mean? I didn't volunteer. I didn't have internships in science settings because I was paying for it and saving money for the school year, right? So for all of us to be able to realize as people in corporate settings, take that into account, interview someone, get to what kind of worker they are and team worker they are, and work experience sometime is just as good as an internship at a scientific company, because I was a waitress and a bartender, and you learn to be nice to people and not flip out at work and all these things that being at work all these years you see happen with people. So I think kind of for us all just to broaden our horizons and realize we don't know what we're looking for. You just ask for kind of a diverse pool of qualified candidates, And then, you know, you got to kind of take them as they come, and we all come from different places. That doesn't mean one person's going to be a better employee over another, you know. And I think that mentorship with middle school and high school students, it'll kind of like open that up for all of us.
Mhm.
This is fantastic. Really appreciate your thoughtfulness. We're so happy that there's a partnership between AbbVie and the Worcester Public Schools, and hopeful that students will be in school soon and things will be able to resume as they were. It becomes so much more challenging at a time like this to keep those kind of community engagement partnerships alive and well. So thank you both so much for your perspective. We're going to shift now to hear from some high school students who, well, no longer, not everyone on it is high school, but who as high school students participated in the state science fair. And so I'm going to hand it over to Catherine Zhang and she'll introduce herself. Thank you so much, Jen. Thank you, Lynn.
I'm gonna share my screen. All right. Hi, everyone. I wish you're doing well during this time period. I'm Catherine, class of 2020 at WPI, and I'm currently pursuing computer science and mathematical science double major. So the project I'm presenting to you all today is from last year's science fair, which I conducted during my senior year at Films Academy. And my mentors are from Woto Oceanographic Institute. So my project was about visual function of AHR1B in embryonic Daniel, which is zebrafish. As you can see, it's a picture of zebrafish here. So first, I think I should explain what is AHR1B. So let's start with AHR. The full name is aryl hydrocarbon receptor, which is a ligand-activated transcription factor that is involved in xenobiotic metabolism and detoxification, which is its one-on function. So AHR plays a very important role in human bodies, preventing those harmful chemicals from accumulating in your bodies to protect you. So while humans, we only have one AHR gene, zebrafish, they have 3 AHR genes. So AHR1A, AHR1B, and AHR2. So AHR1B is the one I studied here. So where does the idea of this project came from? Why I studied AHR1B? That's because a previous study done by my mentor, Dr. Han, has determined that AHR1B protein localized mainly in the developing eye during embryo larva stage of zebrafish development. So it suggests that AHR1B might play an important role in vision function. So that's why I try to find more evidence suggesting that a relationship exists between AHR1B and vision. So my hypothesis was that embryonic zebrafish in which AHR1B was knocked out would show inferior visual ability compared to the control group. So the assay in the experiment I used is called optomotor response assay to test the larvae's motion detection ability following the movement inductions. So here is like setup of the experiment. There are 10 wells in each agar plate. And if you can see here, those tiny little dots with a tail, those are the zebrafish larvae. They are 7 days old post-fertilization. And if you can see, there are like striped shadows on the plate, and those are red stripes. You can see the color here because I took a screenshot on the monitor. And those red stripes, they will start to move in the experiment. So during each experiment, a 10-minute video was recorded. For the first 5 minutes, nothing. There's no movement inductions. We just let the fish swim around. And then the red stripes, they will start to move forward, which is toward the forward zone, and then reverse, which is toward the reverse zone for 2.5 minutes each. So ideally, the larvae, they will follow the moving stripes toward the end of the well and then move back and forth, trying to follow the stripes and towards the end of the well again. So I calculated the fraction of distance traveled in forward and reverse zones, which are here represented in the green and orange, over the total distance traveled in the whole well during a 30-second interval to represent the activity level of the zebrafish in that zone. And here is my result, the graph. As you can see over here, during forward movement induction, the activity level of the control group, which is the blue curve, is higher than the mutant curve, which is the pink one. And also there's a distinct difference in the control group when the induction happens or the change direction from forward to reverse. As you can see here in the blue curve and also in the brown curve. But compared to that, as you can see here, the mutant group, they show the milder change. As you can see, it's the pink curve and the yellow curve. So these results show the trend that the control group appear to be more sensitive to the inductions in the mutant group, which might suggest that the control had a better visual ability than the mutants. And there are also 2 more studies on the same gene in fruit flies and mice that suggest the same idea. Knockout of those genes result in vision degeneration in those 2 species. So mice with AHR knocked out, they developed aged macular degeneration like pathology. And fruit fly with spineless knocked out, which is spineless is the ortholog of AHR, essentially means the same gene, but just in different species. And those mutant fruit flies experienced degeneration in their color vision. But there's a very interesting finding in a study. In the spineless mutant fruit flies, according to study, overexpression of the spineless, the gene, can induce the photoreceptors in their ommatidia to choose yellow fate. And thus increase color sensitivity, which is the opposite effect of the mutation itself. So I found that it's really cool because maybe overexpression of AHR might be able to do the same thing if it is proved to have a relationship with vision now. So that was the overall idea of my project. There are some limitations. There are also some improvements for further studies. Like I can test the color sensitivity of control and mutant zebrafish instead of the motion detection alone, and see if there's a greater effect, because color vision and motion detection, they're 2 distinct but not completely independent features of vision. And also according to the fruit fly test, there's an effect on the color vision of fruit fly. So maybe there's also a more obvious effect in the color vision in zebrafish. I think, but still the exact role of AHR remains unclear. We need further investigations to see if there is any relationship between the AHR gene and the vision. But there's still a great potential in this study though. Like maybe we can prevent or even treat some eye disease using the, according to the overexpression of the spineless in fruit flies, maybe we can do some, treat some eye disease or prevent them. So just wrap it up. I think science fair was a really, really precious experience I have. And also just based on Jane's story and Lane's story, their experience, I was like so moved because I feel like I'm so grateful that I had the opportunity to do everything, to work in a lab, to use some cool technologies like PCR, to work with mentors, like teamwork, and also got experience, especially this project is in an unknown area in science, which I feel like it's very nerdy, sounds very nerdy, but I think it is the charm of science. It's always those unknown areas. So that's the end of my talk. Are there any questions?
Thanks, Cather. That was tremendous. Can you speak a little bit about those resources that you had that were able to support you in your research and that access to the lab? And I know you have some, based on where you went to school, there were some resources as well in the community.
Yes, so Falmouth Academy is a high school that located right on Cape Cod, which is really right next to a lot of labs like Hui, which is the place I worked in, and also MBL, which is another, the marine biology lab, which is also another lab. And a lot of students, the school has a relationship with those labs. So a lot of students have the opportunities to find a mentor through one of the teachers in our school. So she will help us to find mentors and even find mentors outside of this, those connections, just trying to help students out. And science fair is actually one of the required thing we have to do for in our school, like from 7th grade to 11th grade actually. So as a senior, you can choose to do major major effort, which is not science fair, which is like something you can develop a topic, like anything you want to do. So history, range from history to science, and from even sports, you can do those. So I think that's like, I feel really grateful that my school did this. So I had the opportunity to do everything, everything like that. It was really cool. And I was actually the first time when I moved to Cape Cod when I was a sophomore. So those was the first time I ever do a science fair. Have to write a paper, have to give a speech, have to talk to the judges. So I think that's really cool. Helped me to prepare for some college experience, like giving a speech and do the same thing. So I'm really happy about it.
Thanks. Actually, it would be great for you to speak a little bit about that part of what else aside from the research is part of a science fair project. I think we often forget that it's the whole package, right? The research is what sort of is the longitudinal work throughout the year. And then there's the final presentation piece, which requires a whole separate set of skills, but are equally important in some ways to communicate your ideas. Can you talk a little bit about that experience?
Oh, so it was really hard at first. I had no idea how to write science as like scientific language, especially English, because it's my second language. And I have to practice over and over again just to keep my talk make sense, I guess. But also like making the board part helped me a lot because I can follow along with everything, showing all the graphics and all the data I have and some pinpoints I have to talk about. Well, this year it was especially hard without the board talking to the judge, but it's just a lot of editing, a lot of writing, a lot of research, because whatever you write on the paper, you have essentially, I feel like I have to have a reference. Did a lot of research just to make sure everything I wrote out was correct. So that also helped me develop some of the skills in doing research and finding the stuff I need, especially like citation and going and looking into libraries and database, a lot of things like that.
Great, thanks. And I want to just tell everyone who's on the call that if you have questions, please put them in the chat and we'd be happy to ask them. So has any of your research translated into anything you're studying now or interested in studying, or are you separating out the science fair experience from college?
Well, I am doing computer science right now, so it feels like maybe not, but I still can do like internship. Oh, I'm actually taking a course. I'm taking a course called GPS, Heal the World, which is essentially we work on practical problems, doing research on it. And this term, we're actually gonna try to interview some of the experts in the area. So my team, we're working on meningitis in southern Nigeria, because it is they're on the meningitis belt, which is they have the highest case ratio, like rate in the world. And we were still like looking into all those papers and what is meningitis, are there any treatment for it right now, and what is the current treatment. And not only actually in science part, there are also like some social implications, like their economies, and just a lot of things kind of intervene with each other. So we have to find those informations and do studies on them this term, hopefully, and trying to find a solution, like a potential solution for it.
That's great, thanks. We do have one question here too. Was there one time when you thought you had solved it? This goes back to your science fair project. Can you describe that moment?
Oh, just when I analyzed all my data through the software, and when I put in all the data and generate my graph, I was like, ooh, that looks something interesting. So I was pretty happy, but still, because my project, all my data was behavior assay, so there's still like a lot of large variation between my data and data. So I use standard error instead of standard deviation. But still, one of the I was saying my data was not significantly statistically like significant. But one of the judge at my school science fair, he was saying, when I look at the data, I think you can be more confident than that. He said your data actually represents a stronger idea than what you just said. So I feel like maybe, but still we don't know exact role. We need more further studies into that.
Great, thank you. So I think it's time for us to move on to another student. Thank you so much, Cather.
Welcome.
I'm going to pass the mic to Arnav Mishra, and I will give you the floor.
Thank you so much. I will just share my screen real quick. There we are. So, hi guys, my name is Arna Mishra. I am currently a student at the Massachusetts Academy of Math and Science. So I'm a junior there. And this was my science fair project from last year, my sophomore year, where basically I made a technologically advanced cane to help the visually impaired. And so the name of it was Helping the Deaf and Blind One Step at a Time. And so I've actually been working on this project since my freshman year of high school and decided to do a continuation moving forward into sophomore year. And so before I started this project, I actually visited India for a little bit of time. And so what I noticed there was that where I was living, we didn't really have that much money to get a backup generator. And so there used to be these frequent power outages. And so we really had to navigate in the dark if we really wanted to move around in the nighttime or even in the day sometimes. And my grandparents who were living there had a really hard time just getting around. And especially considering that they were in the older population, it could be fatal if they were able to bump into a certain object. And that got me thinking, what about the people who have to live in that same darkness for the rest of their lives? And according to the World Health Organization in 2010, more than 285 million people have to go through that. And those are the visually impaired or the blind. So my problem statement for this project was, with millions of people around the globe not being able to see, I wanted to try to create a solution to help the visually impaired be able to walk a little bit more independently and safely. And so my hypothesis for this project was I was basically creating a cane which used sensors and vibration modules. And so how it worked was that the sensors would basically detect where an object was in relative position to the user. And the vibration modules would vibrate at a certain frequency. And so as you got closer and closer to an object, it vibrated faster and faster. So my hypothesis was simply that if an object is placed in the peripheral vision of the user, the cane will be able to detect the object's relative position, no matter the material used, because I wanted to make sure that this worked with any type of material objects. And I did this using the infrared and ultrasonic sensor. And so here are just two diagrams of the different hardware components that I use, the Arduino Mega and the ultrasonic sensor. And so what the Arduino Mega basically is that it's an electronic source board where you can take any programming that you have onto a platform called Arduino IDE, which has a programming language similar to C++ or Java. And so what you're able to do is that you can program what you want the hardware to do, and that's all connected through this board right here. So this board would be connected to my sensors that I was using, the vibration modules, all the wires and components that I was using. And the beauty of it is that once you upload your program from the Arduino IDE into this board, it can run independently. So it does not require any Bluetooth or Wi-Fi, meaning that this cane could be working anywhere in the world. So that was a really interesting point that I saw. And so the ultrasonic sensor right here is just a sensor that I used. It basically detects or can say how far away an object is by emitting sound, so using echolocation. And it basically tries to figure out how much time it takes for the sound to come back to the sensor. And once it does that, it converts that time into distance using a time to distance formula. And the infrared sensor works in the same way. However, rather than using sound waves, it uses light. And so we will dive into that a little later. So here's just my procedure and materials needed. And so I wanted to test my cane and then my prototype on wood, cloth, glass, and aluminum to make sure that it would be able to work with the most commonly known materials in our world today. I used 4 ultrasonic sensors and 1 infrared sensor, which I'll explain why later. And everything else was just to set up for the experimentation. And so right here is my experimental design and design criteria. And so I had 3 phases for my testing, and I had more than 1,000 trials in total to make sure that this could work in a variety of situations. And so the first part is where I basically put the sensor, I was using ultrasonic or infrared, onto an elevated plane. And I put a different material object, wood, aluminum, glass, and cloth, at a certain distance away from that sensor. And so let's say I took aluminum. I would put the aluminum 50 centimeters away from either one of these sensors, and I would test whether or not the sensor was accurately giving me the information saying that that object was 50 centimeters away. And so I can take that for cloth, for example. I can say I wanted to test the cloth from an ultrasonic sensor perspective and put that cloth 200 centimeters away from the ultrasonic sensor, and then tested whether or not that ultrasonic sensor was giving the output that that object was 200 centimeters away. And so that was the first part. And so in my testing, I found out that the sensors do not work with all objects independently. And so I wanted to make sure that they would obviously work together in a sense that they could cross-reference their information to get all material objects. And so the second part is where I tested the ultrasonic sensor and infrared sensor working together to cross-reference their information and to make valuable and reliable decisions on how far away an object is. And so this would basically be working with all the objects present. And so in my testing, I figured out that the ultrasonic sensor, for example, worked with all the objects except cloth. And this was because when the ultrasonic sensor is emitting sound, the cloth is sound dampening. And so because of that, the frequency of the sound goes to the object, but only stays there. It doesn't come back to the sensor. So the sensor cannot detect whether an object is there or not. And the same thing is for infrared sensor to glass. Because the infrared sensor gives off light, the light goes straight through the glass. So if you have a window, the infrared sensor would simply just not work. And so using those 2 sensors together, I wanted to make sure they could detect all the objects there. And so my last part was where I basically just connected the vibration modules to the sensors given to make sure that the vibration modules were using that data from the sensors to vibrate at a certain frequency. So here's my initial prototype. I used 4 ultrasonic sensor, 1 infrared sensor. The 1 ultrasonic sensor, infrared sensor would be pointed straight forward to get any object in front. There would be 2 ultrasonic sensors, 1 to the left and 1 to the right to get the full 180-degree peripheral vision. And then I had 1 ultrasonic sensor actually tilted upward. And this was because I wanted to make sure it could detect tabletops and tree branches, things that wouldn't be able to detect with just a sensor positioned straight forward. And so for my experimentation, again, there were more than 1,000 trials done, but it's totally worth it in the end. And what I noticed was, again, that the infrared sensor did not really work with the glass. It was giving always 30 centimeters because that was its blind spot. But otherwise, everything else worked great for the infrared and ultrasonic sensor. You can see in the graph right here that while it is 30 centimeters, the actual distance is 30 centimeters. You can see that in all the testing done, the ultrasonic sensors and infrared sensors were saying that, oh, that object is 300 centimeters away. And so I actually voided data from the ultrasonic sensor detection of cloth in this graph right here. And that's simply because the ultrasonic sensor on cloth was giving data values of 2,000 centimeters when it should be like 200 or 150. But that just turned out to be an error code. And if I had put that in this graph right here, the whole data would be skewed and you wouldn't really be able to see that relation. And so my second graph right here is a vibration module working depending on what the ultrasonic sensor is saying the distance is. And so overall, the vibration modules were working pretty coherently with the ultrasonic sensors. And so you can see right here that the graphs match up to where they should be with 30 vibrations. Mostly the vibration modules were giving 30 vibrations, and the same goes for throughout the testing. And so here's just some pictures of my cane right here. You can see the vibration modules are at the top. So they would each be corresponding with their respective ultrasonic sensor. You can see the Arduino in the first picture, the heavy amount of wires used, and the sensors in the second picture. And so here's my bibliography. Thank you so much for listening. Do you guys have any questions?
Thank you. Can you talk a little bit, Arnav, about what, so I think what we find with student projects is that there's just so much that you've covered and had to deal with, and so many, especially in your project, different, there's there are different technologies that are going into this. Can you talk a little bit about how much of this was stuff you learned in school and how much of this was independent interest in what you did on your own?
Yeah, sure. So actually, before starting this whole thing in freshman year, I had no experience in programming, so I had to learn all of that myself. So in my school itself, I was basically a freshman and sophomore, so we couldn't take really advanced computer science classes or advanced engineering classes. So most of the stuff you did have to self-learn. However, the resources were all there. You could obviously ask one of the teachers at Shrewsbury High School to help you out, such as the engineering department or computer science department. But for me personally, I was able to look at a lot of databases, do a lot of research on the programming and engineering parts, and that's how I got my project.
And can you share what role science fair plays at the school that you were in when you did this project? Is it required? Is it optional? There are different ways in which students come to it, so it'd be helpful to know your school situation.
Yeah, absolutely. So when I came to Shrewsbury High School from 8th grade, there was an option to take this class called Research Methods. And so it was completely optional, but it was notified as the highest class you could take. So obviously a lot of people wanted that challenge. And so this Research Methods class that they had was basically where they inputted the actual science for into the grade or whatever science class you were taking, whether that's biology or chemistry. And so once you were in that class, you were required to do the science fair, but the teachers would help you along the way, kind of setting up all the deadlines on when to get your research paper out there, when to get your data analysis done, when to get the conclusion done. And it was all connected to the school fair that we had.
Great. And to tie it back a little bit to our earlier conversation about volunteers from the community in the classroom, did your school or did your class have any experience with outside resources, either as volunteers or people that sort of supported different students on their projects?
So what happened at our school was that we weren't connected to any organization in particular, but what you could do was that there were some labs available at the school that you could use, but the science fair teacher would have to set you up with certain departments. So if you're looking at a lab at, let's say, UMass Amherst, which some people did, you would have to contact them specifically, but there wasn't really some organizations in particular that Shrewsbury was really close with. So you just had to look for outside information.
We have a question about how much heavier is your cane compared to a normal cane?
So what I wanted to use was that I wanted to get a balance between a white cane and a structural cane. And so a white cane is basically just like a slim rod. It weighs almost nothing, where you can basically feel around for objects. But this cane was implied for the older population, for my grandparents in particular. So I also wanted to make it a structural cane. But this thing probably weighed around, I would say, 5 pounds. It's not too heavy, but it also is a structural cane. So when you're walking around, you don't have to basically put it at an angle, because the sensors will basically do that for you. So you'd use it as a regular structural cane, which is already pretty heavy, but the sensors did not add much weight to it. It's just the cane itself.
And I think you mentioned that you'll be enhancing this and continuing to work on this next year. Is that correct?
Yes, so I've been working on this since my freshman year. I worked on my sophomore year, and now in my junior year, I want to advance the cane into making it more like basically in the real world, viable in the real world. And so I'll be working on that with my science fair teacher at Massachusetts Academy of Math and Science, and trying to develop that even further.
Well, wonderful. I think we need to wrap it up now. Thank you so much, Arnav. It was great to hear about your research. And I just want to close and thank everyone for participating today. It's such an interest. I think it just adds so much color to the process to hear about all the different perspectives and to hear about that pathway between middle school and high school as well. And I'd love to see us working with more middle school students who can then transfer this this work into their high school experience. So thank you so much, everyone. I want to thank WPI for their continued support. And I also want to thank Hanover Insurance, who has been a supporter of the Region 2 Fair for many years as well. And to all of the volunteers, the teachers, the judges, there's just such a host, sort of an ecosystem that makes this all possible. And for that, we're very grateful. If anybody has questions, you can always find us at scifair.com or email us at info@scifair.com. Thank you so much for participating today. Bye.
Thank you, Helen. Thank you. Nice job. Thanks.
Thanks so much.